In the prior to this present time, when you went in for a surgery, the strategy was just pump glucose, right, to keep you from going into hypoglycemia during anesthesia. And the surgery can take a long time, and you recuperate, and it takes several days and so on. And that's not done now at the state-of-the-art hospitals. Okay, so you can see, thanks for revealing your medical information to us here. Okay.
Oh, I can reveal more.
Oh, well, all I'm saying, I use that to make a big point that those discrete measurements, sort of randomly picked, someplace in there where you take a measurement, right? It shows that exact minute, but it doesn't really tell the whole story. Just to belabor a point. So this is what you did today, probably, huh? Something like this.
Use the lancet, yeah.
Stab, little blood.
Yeah.
So we get letters from people all the time, lay people that go on the web and find our work and so on, and then they write a letter. So the most common one is something like some mother will write a letter and say, "Oh, my daughter just turned 10 years old today, we're having a big birthday party. And incidentally, she just had her 10,000th finger stick today." Okay? And that's kind of the anti-birthday party, they have a special different kind of cake thing for that, right? 10-year-old kid. And of course, that's hardly useful because that means she must have stabbed herself about 4 times a day for the last 10 years, approximately. And, just pick 4 of those points in there and say, okay, that's where we hit it, right? Doesn't really tell much. Now, I can see other things like cholesterol. It doesn't vary, it's not going to go up and down like this in the middle of the day, right? And so there are plenty of other things where you take a blood sample and it's pretty reflective of where you are, But, so this is the existing technology. Now, I wanted to, before we got into the glucose monitoring part, which is the big story I want to tell, I wanted to talk about other therapies that are underway, okay? Other approaches to diabetes therapy. And let's see, what I was going to do here is show you a certain website which you can go to that has the National Diabetes Plan, okay? And we're going to look at this plan. Let's see if I can find it easily. It's called the Diabetes Strategic Research Plan. So the reason, and so I'd like you to look at this at your spare time, after class sometime. Go to this site here, look up this, and the site's on your notes there, so look at this plan. This plan is hot off the press. It's just finished this year. And, this is a useful thing for people who are developing a medical device. You want to know everything in the field. You want to know, is my approach going to be the real useful approach? What are the alternative approaches? And what are people thinking and all that stuff? So in this national so-called Diabetes Research Strategic Plan, the final plan here, it's got sections that are pretty informative about diabetes. Okay, so you're going to want to find these sections about your medical device approach. Okay, something similar to this. And for example, I'd like you to look at this section, the beta cell. Okay, and there's one here called Bioengineering Approaches for the Development of Arch Pancreas to Improve Management of Glycemia. And then plenty of other, so this is a big report here, and a lot of it bemoans the problems of diabetes. It says, oh, here's all the things that go wrong, etc. But these 2 chapters in particular are things that engineers find appealing because they're all about how we're going to fix this problem, okay? Most of the others are just, okay, here's what we need to do with more research to figure out what's going on, actually. But then there's a couple there that are engineering oriented. So, so this is an example. When you make a medical device, whichever is your favorite device, you've got to find out some national perspective, okay? And of course, you know, these plans, you know, so I wrote a chunk of this plan, and I got to tell you, it took many rounds and lots of, you know, I'd write something and then they'd send it to a whole bunch of people and say, oh no, that word shouldn't be there, or this word ought to be added here, you know, that kind of thing, right? So I'm telling you, this plan took 2 years to write, okay? And okay, so it's pretty sanitized, you know. I mean, it's there's a lot of opinions in here, but the ones that are on the edge are kind of not on there now, right? And the ones that are mainline and pretty clear, widely accepted, this hasn't. Okay. So you're going to want to do this for your project. Okay, back to where I was here. Now, before, you know, going on to this story here, in fact, I'm going to turn it off just so it won't be too irritating. Let's see if I can do that really. I want to talk about some of the options for medical devices that are presented in the plan, okay, or that people have been working on for 30 years, basically, right? And just to give you an idea of the alternatives. So I don't happen to be working on most of these. I do work on some of these, but you know, I'm just bringing this up because you know, you want to consider what's going to be the easiest way to get to the target of you know, addressing something to do with diabetes. So that's the plan now. The idea is that so I'm looking at the top of this session 2 part here. I mentioned that one strategy that's been proposed and worked on for quite a while, at least 40 years I know of, is transplantation. Okay? So, well, how does that work? You obtain a pancreas from a donor. Usually, of course, that means since you only have one pancreas, not like we have 2 lungs and 2 kidneys, and you know, we have spare parts in some cases, but not in the pancreas. So you get a pancreas from a donor, it's got to be a cadaver donor, okay? Now, people have tried, well, what if we just take 1/3 of the pancreas of a healthy person and we'll transplant it? Okay, that works a little bit too, but, you know, the volume of pancreatic material may not be adequate to actually produce enough insulin to control blood glucose, so they don't really use that strategy much anymore. So, you know, this is a common thing. Now, I'm sure you're aware of the problem with transplantation is we have this immune system that Mother Nature set up a long time ago to thwart the transplant surgeons, basically. I don't know what other, well, I guess it keeps all the bacteria from taking us over, all those other nasty things, vigilance on cancer and stuff, right? So there's plenty of good reasons for it, but one thing it does, it makes it really hard to find a donor and a recipient match, okay? And so there's a national bank of organ donors, and you can get registered on this thing, you know. And so what they do is they draw some blood and they characterize receptors on your white cells, and you know, there's a big variety of possible receptors and on their white cells, and they have this so-called major histocompatibility complex, which you can identify it, okay? And then they look at the person who needs the organ, and of course that person's had their receptors all characterized, and they're pretty well understood and everything. So the question is, how close can we get to a match, you know? Well, the chances of finding a match are, you know, like 1 in a billion, right? If you're not related in any way, there's probably somebody else somewhere on the planet that might be a pretty good match, you know? But chances are pretty doggone small. And of course that person may not just want to give you a piece of their pancreas, you know? Or be dying at the moment you happen to need a pancreas, you know? So that's one of the big problems, is how do you get enough organs to transplant? Well, the story is a little different for people who are close relatives. You know, it's common that in, you know, a family of say 5 kids or something like that, maybe 2 of them match each other pretty well. because their parents provide. And of course, you can have another situation where two in that family are completely opposite, and there's no way they match in any way. They might even look the same and everything, but when you actually test their white cells, that's not going to work. So anyway, this is the part of the story. So there's this national database of just going to get rid of this for the time being, that lists all of this stuff. And I've often been a big advocate of people getting signed up. If you're ever thinking about donating kidney or part of your liver or something like that, I think it's a good idea to get characterized. Right? All my data's on there, and people in my family are on there. But the chances of finding a match are pretty doggone small. Now, the problem in diabetes is really different. So some years ago, decades ago, they said, okay, well, there are cases where there are identical twins, one of which has diabetes and the other one which does not. Or there's a couple of cases of triplets that are known where one of the triplets is diabetic, right? Type 1 diabetic, and the other two aren't, and this kind of thing. Okay, so maybe we can take 1/3 of the pancreas from the healthy twin and stick it in the diabetic twin. Okay. That doesn't work at all. It doesn't work in the slightest bit, because diabetes is an autoimmune disease, and it's very discouraging. What happens is they've tried this in 2 cases before they realized, this is. So what happens is you transplant the pancreas into the diabetic twin, and then suddenly the antibodies start flaring up, and this whole process of white cells start taking out the new pancreas, right? Just like it did before with the old pancreas. So autoimmune disease is not a great candidate for transplantation. If you get the most perfect match possible, there's a disincentive to that, because then the diabetes process will wipe out the implanted tissues. If you get a vastly different match, there's a disincentive for that, because there's a normal transplant rejection process that goes on for mismatches. So we're stuck, basically. So that was discovered in about 1970, and it was a pretty big surprise and big shock, basically. Now, since then, the story's actually gotten better, because there have been a number of drugs introduced for transplant rejection. So that means you don't have to find the other billion in the population that matches you exactly. You can get a transplant that's actually pretty close. You still want to get as close as you can in matching all this stuff, right? But if you're willing to take anti-rejection medicines for a lifetime, then you're in business, right? And there are a few of them, okay, like a handful. And these work more or less well. It's there's some possibilities, and there's a tremendous amount of work in the pharmaceutical industry to find the next one, right? Yeah. The problem with those medicines is that they, of course, have side effects, and one of the side effects of a group of them is kidney failure. Most pills can attack your kidney if you give enough doses of it long enough and all that kind of thing. So kidney failure is an outcome, and kidney failure is the outcome of diabetes, so without being treated, right? And so if you get a transplant that now requires you to create a controlled kidney failure over the next 5 or so years, that's not thought of as a great idea. Just swapping one disease for another. You don't have diabetes anymore because that's taken care of now, but wait a minute, now I'm a kidney failure subject, so, which is as bad as diabetes. So, that idea isn't so popular. Patients aren't just excited about that at all. Now there was an advance called the Edmonton Protocol. It came out about 10 years ago or so. And Edmonton group in the hospital there in Edmonton, Canada, cooked up a mixture of a cocktail of these anti-rejection medicines that is an improvement. Okay, it's whereas the previous treatments led to kidney failure within 5 years, this Edmonton protocol is probably going to get you a much longer time. Okay, we don't know yet. I mean, it could be 20 years or something like that before you get into serious kidney failure. So the Edmonton protocol. So that's that may allow. So that may reduce the immunity barrier for transplantation or minimize it. I emphasize that, you know, there are plenty of people getting heart transplants, liver transplants, all these other kinds of transplants. I shouldn't say plenty, but there are around 10,000 a year in the U.S. That have the same problem, basically. It's just the pancreas is so much more of a fussy organ than the heart is, for example. And so this matching business and all this kind of thing is turns out to be acutely important in the case of pancreas transplantation. Whereas you can kind of get by, you know, as best you can with a heart transplant and, you know, that kind of thing. But so that may reduce the, you know, some of the concerns about these long-term medications. But then you still love this problem. Well, where do we get these pancreases, you know? There's claimed to be like 26 million Americans in the U.S. who are diabetics. Okay, well, wait a minute. There's not 26 million people dying every year who's willing to give up their pancreas. You know, there's maybe 10,000 people who, you know, drive their motorcycle into a tree and there's brain dead, but, you know, you can still rescue some organs from these people, right, if they are willing, right? It's on the order of 10,000 in the U.S. a year. But it's not, you know, it's an order of magnitude on this whole thing. Well, so there are some very creative solutions to this problem in the wings, or potential solutions. Okay. One is, well, why don't we grow a colony of pigs? Okay, and before these pigs are born, we'll substitute the gene of the person who needs the pancreas in the pig, right? You can introduce genes various ways into animals in some limited fashion, right? And so that pig, when it actually gets born, is going to look genetically and immunologically pretty much like this person who needs its organs later.
So it'll actually have your MHC.
What's that?
It'll actually have your MHC.
That's the plan. I'm not saying this is. I'm on the edge of science fiction here. Okay. The plan is to grow these pigs, but before they're born, you re-engineer their immune system entirely to look just like the person who needs the help, right? So it's a serious and a very exciting possibility, actually. Of course, then the pig grows, and once it gets the right size, then it becomes a donor, right? So there's a significant industry about you know, trying to figure out how to do this and so on. I think it's an interesting prospect. There is one significant issue with it, and that is the possibility of cross-species contagion of various viruses and so on. Each species somehow has come to terms with a set of viruses we have in us, right? But they're not taking us over at the moment, right? We sort of come to a truce, you know, that our immune system can fight those off, you know? And we have our special ones, and a different species has its special ones, okay? And if you succeed in crossing the immune barrier and transplanting an organ from one species, from a pig into a human, you're going to transplant all the virus burden that pig carries with it into the human, okay? Well, much of that may be inconsequential, you know, but then there's a high possibility there's something in there that pigs have dealt with ages long ago, but that we are totally sensitive to and could wipe us out, right? And of course, then it might be transferred from human to human. Once you get a cross species, then human to human transfer, you know, that happens all the time, right? So there's a big concern in doing this. In fact, Congress got involved a few years ago and said, okay, we're not going to do that now until we somehow clean up the genome of the pig, right? And then somebody else said, well, you totally clean up the genome of the pig, our genome is handled a little bit differently at the detail level for everything, and maybe there's something that is still can create a problem. So there are companies set up to do this, their investment, you know, and everything like that going, but it isn't so simple, you know. It has potential and it's exciting and all that, but it also has potential for, you know, like we have these SARS virus, you know, there's some lemur in China or something that, you know, transfers this, and they're totally healthy, but then people eat that, suddenly they're having all kinds of nasty virus infections and stuff, right? Serious. So that we don't want that cross-species contamination issue. So, you know, that's okay. So that, I'm just telling you all the modern stuff. People are working on this, okay? There's a need for solving these kind of problems, and they're pretty well-defined problems at this point. So transplantation. Now, there have been clinical trials, excuse me, in transplantation, where people are willing to go on the long-term anti-rejection medicines, but the results still have been pretty dismal. There's a 4% survival at the end of 1 year. There's been now 5 trials done. Okay.
This is for a partial.
Partial or cadaver, yeah.
5%.
5, yeah, on the 5, never hit 6% yet. So, you know, that's still an issue. I mean, just put it in perspective still, okay? That's medicines, okay? And, okay, well, that doesn't sound good, but, you know, you still got to keep working on this, because you can't guarantee that the alternatives, which I'm going to be telling you about, are going to be that much better. So, anyway, there's a big team of people working on transplantation, is the message I'm trying to give. And there's plenty of things to do in that area. Now, so okay, plan B is, instead of transplanting a whole pancreatic organ with all that, you know, protein producing capability, 99% of the volume of the organ, really not what we want anyway, but forget that. Let's go for the beta cells themselves, or the islets, right? So you can take islets out of the pancreas and, you know, effectively clean it up potentially. And maybe you can take those islets and implant them, inject them, or put them into a recipient. And this is a strategy that people have worked on for at least 30 years. The guy in the next lab, when I was a postdoc, Bill Chick, he's the guy that first did this, and shows that works just fine in dogs. You know, you can take dog islets, and this was 1976. He injected them into another dog who was diabetic, right? Yeah, they work fine, you know, to a certain level, right? They eventually are, you know, degraded by the immune system, but that could be over a period of months or years or something. So then the idea was, okay, once they're degraded though, you become immunized against those islets, so you can't get a second dose of those, you know. Not from the identical same person, but probably, you know, it depends on what the immune response was to. If it's some generic part of the islet that everybody has, that means you're not going to get an islet again that works, you know. Or if it's highly specific immune response to that one person's islet, maybe then you can get another person's islets and try them out, right? But, you know, these are kind of like reduced options, you know, each one of these stages, you get up with fewer and fewer possibilities. So the idea is, well, why don't we immuno-isolate or protect the islets in some fashion? This means what? Well, you can get these islets and maybe you can wrap a synthetic polymer coating around them. And this coating, you know, these synthetic polymers very rarely, if ever, raise antibodies. You can't really do that to synthetic polymers, usually. And they could serve as a way of protecting the islet, right? And then, you know, maybe you make an islet and it's got this coating around, you make enough of them and you just inject that, or you put them into like a little soda straw and you insert the soda straw into your peritoneal cavity, you know, so you can get it back out again later. Various strategies like that. And that's been worked on for a long time. And, you know, that's actually pretty tough to do because you have to, there's many reasons. One is you have to tailor the polymer coating so that glucose can get in there, okay? And when the islets start making insulin, that's got to get out of the coating, right, into the body. And insulin is a molecular weight of about 5,600, okay, and glucose is about 180, right? So insulin is not a really big protein, but it means you got to have a hole in those polymer coatings. It's big enough for a 5,600 molecule to get through. Well, there's plenty of nasty molecules that white cells make to kill cells that are around the order of 1,000, you know? So, yeah, so that could still be an issue. You know, if you get a white cell that's going to embrace that encapsulated islet, they're going to make hydrogen peroxide typically, which is, you know, molecular weight of 30 or something like that, right? And that kills cells pretty well. You know, that's one of the strategies white cells use to kill bacteria, is just get real close and give them a big hug and then deposit some hydrogen peroxide on them, right? And that's the end of that, right? That's the way it works, actually. So, I mean, so, okay, I'm just saying, it gets to be pretty hard to imagine how you're going to really make this polymer barrier that could be totally effective. And then, you know, white cells have this other strategy. If they can't really inactivate or eliminate something they think is foreign, they form a halo around the object, right? There's a whole bunch of white cells and they form a, you know, they'll sort of shoulder to shoulder and just form a halo in the tissue around the object and starve it, okay? So, okay, you gotta sort of think about these, you know, these strategies require some basic understanding of what the body's going to, how it's going to respond. And, you know, of course, you want to try and figure out an engineering approach to counter that or get along with it somehow. So there's an issue. And then the other big issue is the islets in the body are in direct contact with blood vessels, right? So when you need insulin, it's immediately available in the bloodstream. Well, when you take these islets out, coat them some way, put them in, say, in the peritoneum or inject them into the liver, wherever they're going to go, they aren't in contact with the bloodstream, right? They're going to be stuck in the tissue somewhere. And so you're going to have this diffusional distance from the nearest blood vessels to the object there, both ways. When it makes insulin, the same thing has to happen. So we look back at Bill Chick's data in the dog, okay? It was very successful. You know, you get dogs that are pretty well matched, you know, in transplant back in 1976. It turns out those devices just oozed insulin continuously, okay, as long as they were working. They didn't do this response to dynamically at all, okay? That's pretty clear. And, you know, it's just like a slow dose insulin injection, basically. Slow delivery kind of thing. So, okay, you know, it'd be nice to have its own vasculature and everything, you know, rapidly responding and get as close to the natural pancreas as possible, but that's going to be hard to do. So there are groups around the country that are working to make vascular plexus development in sort of synthetic tissue, tissue engineered vascular. And the main focus, one of the big reasons for that is so that you can then somehow connect that with a islet. I mean, so, you know, you can see that the issues are kind of complicated biologically. Now, it's also clear that the islets are very fussy tissues, and when you remove them from the body, if you expose them to air, where the oxygen level is much higher than you're normally happy with in the body, they start metabolizing like crazy, start running like fast, and then a couple days later they stop. Right. And they, you know, they stop just because they either go to sleep or they burn out all their internal stores of energy. And so it took a long time to figure out how can we get these islets to work. I mean, we take them out here and put them in there, but you can't expose them to the air. It's just, that's the end of it, right? The oxygen levels in the body are only about a fourth at the highest compared to the oxygen levels in the room here, right? So when they get this big surge of oxygen, wow, you know, they're going like great, but then that's the end of that. So you think, well, how are we going to process these islets? You know, we're going to get a donor, say a cadaver donor, and we've got to take the islets out of there, and we've got to separate them from the acinar tissue so they won't just get attacked by a bunch of proteins. And then we've got to modify them to make them immune isolated, and then we've got to put them in that person, right? And it'd be great if we had a vascular system handy too, that would be nice. So processing of those islets is a serious chemical engineering issue. Now the other issue is that in order to cure diabetes in the recipient, you're going to need 3 or 4 pancreases. Not just one donor isn't going to make it, because the facts are we all lose islets as we age, you know, or we lose 10% of our islet population. Just normal people who are non-diabetics lose islets, you know, after about age 20. I'm sorry to tell you, you know, okay, your islet supply has been going down, and so is mine. And you know, people with diabetes, of course, maybe it goes down a little too fast, right? All that. But so the point I'm trying to make is that one pancreas for one recipient isn't going to cut it. You're not going to make enough insulin for that. It won't work at all. So you need, the estimate is now maybe 3 to 4 pancreases per recipient.
Or can you get a younger donor's pancreas, like in the early 20s?
That's very reasonable. Can you get a 20-year-old to donate their pancreas, or give you a slice of their pancreas?
There's only the 10,000 car accidents a year, right? So you've got a small population to draw from.
Yeah. You know, they used to not have big enforcement of the helmet law for motorcycle riders, and there would be more organs available in those days. But, you know, I'm not saying that's, we can rely on that strategy, okay? That's a 20-year-old motorcycle rider who forgot his helmet and decides to confront a tree at 60 miles an hour, right? Okay, and might have the pancreas still working after that, right? But that's, yeah. Okay, so I'm making a big joke. There's 26 million diabetics who are sitting there who would like to get this kind of thing. Okay, well, why don't you get these islets and expand them in culture, you know, or get the beta cells out of there and grow them in culture and expand the culture. That's a serious issue, and it's really hard to do. The story is that bacteria, you can expand cultures of bacterial cells, you know, just like crazy. They grow all over the place. They grow over the petri dish, and they, you know, they everywhere. It's great for bacteria, but mammalian cells, it's really hard to expand the culture and get, you know, get them to grow. Of course, you don't want them to become cancers, right, where they're out of control growth and all this. So if you can get some islets or some beta cells and expand the culture, that's another strategy. But, you know, that's been looked at pretty heavily. There's a lot of biologists who have been working on that for a long time. And, you know, I hope they're successful, right? Because that's, you know, each one of these, I'm just saying that these are not new ideas. Some people have been working on them since, you know, the 70s in each case, right? And hopefully they figure out how to do this. And then, of course, you know, you would like to solve the immune problem as well. And people have been working on that for a long, long time. Okay, but anyway, that's the story there. So, yes, please,
So,
please.
But transplantation, would that only be a treatment for type 1 diabetics?
Yeah, that, of course, type 1 are the most difficult ones to deal with, you know, in terms of therapy, right? So, yeah, that's not gonna, what does transplantation do if you're type 2? Nothing really. I mean, because you don't, your receptor is gone, so the more insulin you have, the worse. You know, it's not.
So healthy
it's not gonna,
pancreas doesn't help you.
Yeah, that won't do anything with type 2s. But we're saying, you know, okay, maybe it'd be working possible in type 1s. So now, everything I've said about islet transplantation applies to stem cells. Okay, the stem cell initiative here in California was a big advent. You know, it's all driven by people who had diabetic children, and they were saying, okay, if we can just get stem cells to work, you know, learn how to control them, find them, learn how to control them, you know, and how to get them to expand in culture. That's the big exciting thing about stem cells, and presumably they can make more cells anytime they're in the mood, right? You can figure out how to do this. That was one of the fundamental driving forces of the initiative, the coupled stem cell initiative. That is still exciting and everything, but you know, everything I've said here applies to stem cells, and stem cells have immune antigens on their surface, okay? They're not, so you have the immune problem to deal with. Well, we have some strategies, maybe the immune problem might work, you know, somehow. The stem cell, the problem still is, where is the stem cell in the case of the beta cell, okay? It's not like we know it's someplace in there, but if you ask 5 stem cell researchers, one's going to tell you they think the stem cell is, you know, they have 5 different places where the stem cell. I'm just saying we haven't isolated the stem cells that make beta cells yet. We don't really know exactly where they are and which cell we're talking about. But maybe that'll change quickly, you know, I hope so. But it's not for lack of working on this. So where is the stem cell? Once you find the stem cell, you have to then extract and then learn how to control it, right? This is a big item in stem cells. They have to grow. You can't have them growing out of control because, you know, that would lead to cancer equivalent. And you have to know how to turn the stem cell on and turn the stem cell off. You know, we can't have one kind of tissue in our body that's, you know, we don't want islets just proliferating like crazy in our body, right? And we want to have, yeah, so that means you've got to have some other oral medication would be ideal that you can take and say, okay, now stem cells work, you know, and work for a few days and make lots of islets, and then, okay, take another oral medication and says, okay, now stop and just sit there like many other tissues are doing right now in our body, right? So there's a turn on and a turn off strategy that has to be understood about stem cells. These are basic biology issues. I'm making them as simple as I can understand, okay? And if the biologists were telling me this, it'd be a lot more complicated story. But that's an issue. And then of course you have the immune part of the story for stem cells as well. But maybe there's a way of knocking out the immune response in stem cells. Somehow re-engineering. Now, well, what about genetic therapy? How about this? This was a big item years ago. People were saying, well, we'll just fix the genes, you know, this is going to be the system. And I'm sure most of you realize that's not so easy to do, right? There might be ways of blocking bad genes. For example, siRNA might be a way of, you know, complementing a gene that you don't want, therefore sort of turning it off. But that hasn't been fully understood yet. siRNA is very short-lived, and so that works for a short period of time, but you know, a day later you need another dose of it or something, right? So people are working like crazy trying to figure out how to block genes you don't want. But you know, the problem in diabetes is, you know, there's many genes. It's a multi-gene disease. It's called the geneticist's nightmare, because there's so many genes. These genes are tied into our body weight, you know, they're tied into our metabolic activity levels, and all this, the rest of our maintenance of our general metabolism is directly coupled into the diabetes genes, right? And so some of them need to be turned off in diabetes, some of them need to be turned on. There has to be all this kind of complicated stuff that geneticists, you know, it's a really, really complicated thing. Anyway, so gene therapy, hopefully it'll work, but not so obvious. Now, pharmaceutical strategies. Okay, this is a winner. This is where drug companies make some kind of a pill or injection. For example, there's big interest in industry in modulating the effect of insulin. So many diabetics, when they discover they have diabetes, still have maybe 10 to 20% of their islets in there working okay, but they've lost 80%, okay? And so if you can potentiate the effect of insulin that's produced by that 10% remaining islet base, right? Suppose, okay, it makes insulin. Suppose you have a secondary molecule that you can use to enhance the insulin effect. Okay, this is one strategy. You know, something like that. And of course, there's plenty of interest in making insulins that are slow dose, long lasting. And there's plenty of interest in making insulins that are very rapid, much more rapid than our present insulin. Okay? Because with our present insulin, you can't actually control that spike. Okay? You can't really control it because, well, when it's underway, you give an insulin injection, and the insulin injection won't take place.
It doesn't respond fast enough.
Too slow, too slow. So people are saying, well, what if we have a monitoring system that identifies a spike underway, and we can give an insulin injection that will absorb rapidly and have its effect within a few minutes? So there's a big interest, and you can read about this in that the website I mentioned, in making very fast responding insulins. So that's one strategy, is that just, you know, some kind of there are plenty of other organs in the body that, for example, the liver takes up glucose and makes glycogen into polyglucose form and stores it in your liver. And when your blood glucose goes too low, then the liver puts that back into the bloodstream and it prevents you from becoming hypoglycemic. Okay, well, so we can make the liver, make some drug that affects the liver and adjusts the, you know, fine-tunes the amount of glycogen. And so there's plenty of drug strategies, is what I'm trying to get at, right? And especially for all these complications, people now are looking at, gee, inflammation is a big complication, and that leads to a lot of these problems. How do we manage inflammation? Well, there might be some good drugs for that. And some of these drugs need to have their effects in some tissues, but not in others, okay? So it's pretty sophisticated. But it draws on the incredibly powerful strength of the pharmaceutical industry. You know, that's been just a tremendous American success, right? And so you know, these are things that might be useful. On the other hand, if you pay attention to the news, you'll know also that the number of drugs that are useful in diabetes is being reduced frequently. The armamentarium, for example, is smaller now than it was a year ago, okay? Because some of the drugs have been identified with heart failure over a 10-year period. You know, you get these drugs, you use them for long term, every day. You suddenly realize there's some big complication that was never possible to study before. And so, like a drug like Actos, for example, that affects the liver and was just thought to be the best thing ever by. Suddenly there's a thing in the newspaper that says, okay, Actos off the market today, you know, this is, the study shows that there's an increased incidence of heart failure in Actos users. So I'm saying that instead of okay, this is the strategy of increasing treatment tools, but if you look at the story, actually, the tools are under fire and being reduced all the time. Talk to your standard diabetologist and say, we don't have very many tools. There are very few things I can actually use with confidence that we're sure this is going to create some other big problem. So now I'm going to say one thing and then we'll take a little break. The great strategy is prevention, okay? Who needs this anyway? Let's prevent this disease, okay? So that means weight management, exercise, you know, eating correctly, eating good foods. That's the strategy for prevention or postponement of the disease, right? It doesn't work in type 1s, sadly, okay? But it could work in the other 95% of the population that has type 2. And prevention might help some of the rest of us who are destined to become diabetics, you know, from getting it early in life, or may prevent it entirely. So I think that's the strategy, okay? Now, you young, healthy people, this sounds easy to do, but when you get to be my age, losing weight is a big issue, and maintaining your weight, okay? Just look at the population. People, you know, people are overweight. Me, and others as well, okay? And in fact, the obesity epidemic is growing, okay, not contracting. And it's a really hard one to adjust because it requires people to totally readjust their lifestyle. I mean, it means when you go to the grocery store, you know, I like that, I don't want to have to give up that. Well, you know, that's what causes you to get fat, you know. This is a serious issue, you know, serious. It's great if you can take a pill and fix your disease, but when you're telling people you have to give up your favorite foods, you're gonna have to now get out there and exercise a lot more, you're gonna lose weight, how are you gonna do this, you know? I'm just telling you, that's a really hard thing to do.